So I'm Amanda Brown, glad to be here. Very passionate about training, and this is a fantastic experience that you all are having. And so it's nice to be, further down in the order, let's see, okay, good, of speakers, because the previous speakers from Dr. Nath, Dr. Simpkins, Dr. Karpol that we just had have set the stage, right? Because you're all sitting there, you're wondering, how do I go from where I am now to where I want to be, right? And I remember sitting there in these seats as you are now, looking at, a person like me. We seem to have it all together, and you're like, oh, how do I get there? Okay? And so my journey began. I'm from California originally. I went to UC Riverside, and I had the opportunity, actually, who would have known? I actually came to the NIH back in 1988. You can see how old I am, right? And there I am. And this was an introduction to biomedical research, phase 1. I don't remember all the details, but obviously I was fascinated and engaged, and it had an impact. And so what I'm going to do today, I've kind of interwoven my personal story with the science, okay? So I'm going to give you a background on HIV and tell you how my journey from microbes to the brain started. Okay, I'm going to tell you about HIV neuropathogenesis, talk about how it induces inflammation in the brain, and then some of the approaches that we're using. And so, HIV infection has been with us, it's a worldwide pandemic, for over 30 years, right? We still have no vaccine, no cure. Today, there are over 37 million people living with HIV infection. You can see here the numbers across the world. This is in 2016. More people are living with infection because we do have good therapies, but they have to take those therapies for the remainder of their life, because if they do not, the virus will reemerge. And so these are the number of children newly infected in 2016. So you can still see that across the world, there's still many people being infected every day. And also the deaths are still significant. We have the president's initiative, this was years ago, to bring antiretroviral therapy to places where, particularly sub-Saharan Africa. So that it has had a major impact in extending folks' lives. But, amazingly, well, not in a good way, this is in 2016, right, in the United States. We know how to prevent HIV, right? But we still have people being infected with HIV. Some of these are fueled by use of drugs of abuse. And here are the numbers. So this is still, you can see that the diagnoses from 2010 to 2015, okay, right in those really critical years, people are still being infected. We still have a lot to do on the education side. You can see in the South, in some of our major urban areas, we still have lots of people being diagnosed. This was in 2015 with HIV infection. Okay. I'm going to skip through some of these slides because I wasn't sure how much time I had, so I will be skipping through some slides.
We're actually fine on time.
Oh, we're fine on time. Okay, great. Okay, excellent. And so I always wanted to be an educator, teacher. I didn't hear about research until the end of my senior year in high school. My biology teacher would talk about what he would do if he could go back to school and do research. And so at the University of California, I had the good fortune to meet Dr. Catherine Atkinson. And she is the one that introduced me to yeast biology, and I started learning molecular biology and getting into the lab. So you can see the date there. And in preparing these talks, this is like my second talk that I've been able to give to young folks, I looked up Dr. Atkinson. And she still, in 2016, she moved to Iowa, but she's teaching middle schoolers about Avogadro's number. Okay, so she was passionate about research and science, and so we got our passion from her. During the summers, when I was at University of California, Riverside, I participated in summer research experience programs like you're doing now. And one of them, the first one, was at UC Davis, and it was again, probably through Dr. Atkinson's network, because it was again working with Dr. Chet Price. He was in the Department of Food Science, but also working in the yeast field. And then the following summer, I went to Carnegie Mellon University and worked in the laboratory of Dr. Susan Henry, who was also a yeast biologist. And I got interested, thought I was interested in immunology, microbiology, still was quite an interest. And she said, why don't you go to Albert Einstein College of Medicine? And she was the director of the graduate school previously at Albert Einstein College of Medicine. So with my outstanding grades and scores and letter recommendation from her, I got into Albert Einstein College of Medicine, where I did my graduate studies. And there I got really interested in the host pathogen biology. And I ended up working in the laboratory of Dr. William Jacobs, Jr. When I joined the lab, they had just discovered how to get DNA into mycobacteria. It has a very waxy surface coat, and so they discovered electroporation. We're also using bacteriophages in order to introduce DNA into mycobacteria. And I got interested in how mycobacteria is able to avoid phagolysosomal fusion, and that there could be specific proteins or specific mechanisms to prevent the fusion of the phagosome with the lysosomes, and that protein secretion would be important in this. And so I used genetics, some genetic tricks to actually identify the major secretory protein that would be responsible for secreting general proteins from the cell. And interestingly, it turned out that there were actually two genes that they're very close homologs, but that function in mycobacteria. And this is very interesting. So this was actually the first report of that in a gram-positive bacteria, and subsequently other gram-positive has been also shown in other gram-positive bacteria. So that's one of the things that you can look forward to when you know, as Dr. Nath said, oh, you do the research and then it stays on the shelf. Well, there are people who are looking at the books or the journals, right, on the shelves and build upon your discoveries. And so that can happen. You've also heard from the speakers today about how, you know, what you're learning, maybe you don't see how it might be connected to something in the future, but I was able to translate this Dr. Jacobs' idea of using bacteriophages to report back, say in a sputum sample from a patient, that their tuberculosis is present. And so I actually, I'm going to show you how I use that later during my postdoctoral fellowship. So I moved to the Aaron Diamond AIDS Research Center for my postdoctoral fellowship. And this was a center, so a private research institute, and we had an association with an affiliation with the academic center. At the time, it was NYU, and then the association changed to Rockefeller. And so there I am, and I think I was pregnant at that time, so you can ask me about that later, okay? And so this center was totally focused on HIV infection. There were people doing basic science, translational research. It was at the time where we had just, David Ho had just shown that the triple drug therapy would work, and that you should give that therapy early, and so that you can drive the infection down. So I'm not going to go through the life cycle. You probably know a lot about the HIV life cycle. You can look that up. But I was really focused, again, sort of interested from my my host pathogen interactions on this one so-called accessory protein, but it's actually very important for HIV replication because it's needed. It's called NEF. and it's required for high viral loads. It's a very sticky protein. It interacts with a lot of different proteins and can cause, and it really affects the movement of membranes throughout the cell. And so in terms of the HIV disease course, we know that the virus goes in, right? In a few weeks, you have very high viral loads. The viral loads do come down, but during this period, there's a very robust response of the host to the virus, so that the cells are the T cells, right, are infected with HIV, but then they die, more produce, and you have these cycles, these ongoing cycles, and there's a battle between the virus and the immune system. And in most cases, the virus wins out. You see a decline of your CD4 positive T cells, right? And at this end, this is where we get AIDS. When your T cells drop below 200, then you can get these opportunistic infections. But we know from animal studies and from some unfortunate folks who got infected early and were able to show that there's early seeding of the gut, the lymph nodes, and the brain. And we think that this is an ongoing reservoir and a problem that is still with us. I'm going to skip this one. And so when I was doing my postdoctoral fellowship at the Aaron Diamond Center, my advisor, Dr. Cecilia Chang Mayer, suggested that I focus on HIV infection of macrophages. And that was really a good idea for me because I was really intrigued, by the macrophage. I'm still intrigued by this cell, that it's part of the innate immune system. It's part of our first line of defense against foreign pathogens. And so, but it was difficult to identify infected macrophages in culture. And so I thought of using the HIV, using the GFP reporter gene, fluorescent reporter gene, as a way to, we inserted that into the early gene position in the HIV genome so that when it infects cells, we could see these cells would fluoresce green. So we can use a variety of techniques, cell sorting, to identify these cells. And I got interested in these cells as a reservoir, right? Because the virus doesn't kill the macrophages. And so the virus can persist in the macrophages, which are present in most tissues in the body. And we think that this is an important reservoir. From the Aaron Diamond AIDS Research Center, I ended up at Johns Hopkins University. There was an advertisement in Science magazine to study HIV latent infection of macrophages with Suzanne Gardner. And so you may have seen that, or you will experience that in your career, the papers that you read and you see the scientist's name and then you actually get to meet them. And so I consider her the grandmother of HIV macrophage biology. So it was very exciting to work with her. And when I took the train down from New York to Baltimore, and we spoke, and I told her, I really like to be a faculty member. And so in the Department of Neurology, where I am now, and where where she was, there is a mechanism to move up as a postdoc. You can move up to faculty position. And so I think I'll have a slide on that later. So as I mentioned, HIV can get into the brain, right? And before we had antiretroviral therapy, there was a complex called AIDS dementia complex, and people would exhibit these clinical symptoms: short-term memory loss, mental slowing, reading and comprehension difficulties, apathy, gait disturbances. And so this, and you would see upon neuroimaging these very striking abnormalities in the brain. And although now, especially in the United States, we see less of the HIV-associated dementia, in terms of the neuropathology, we would see encephalitis, reactive astrocytosis, you see myelin pallor, these are microglial nodules, and multinucleated giant cells, because there is, during this inflammatory response and immune activation, you see infiltration of monocytes and macrophages into the brain. And so we say we don't see it very often in the United States anymore, but this is the numbers from 2015. So this is the AIDS prevalence. And so you can see here, we still do have some hot pockets, but there's no region in the United States that remains untouched. And so this is still, if you're going to be on the clinic, you still may see this here in the United States. And so we have, in this era where we do have good antiretroviral therapy, the symptoms are milder. But what we see and what the data shows is that people who are much younger, say in their 50s, are experiencing things that you wouldn't experience until you're in your 70s, 80s. Okay, memory loss, osteoporosis, cardiovascular disease. And so more people are living longer with the infection and experiencing these comorbidities. So HIV-associated neurocognitive disorder, there's a spectrum, right? So with the most severe, but then there's this asymptomatic neurocognitive impairment that is visible upon neuropsychological testing, okay? And it involves any impairment in 2 cognitive domains. And it doesn't have maybe mild effects on daily activities of living, okay? And so why should we care? So there is a report that shows that people with this disorder, it can affect their ability to drive, their ability to remember to take their medicines. They may more likely be a relapse from methamphetamine. And so it could impact their survival. And that's what this study showed. And so where we are now is that people are living longer with the virus, but because it is a reservoir and none of the current antiretroviral medicines actually inhibit the early genes, such as TAT and the NEF protein, which I mentioned, none of the viral factors inhibit viral transcription. And so these early genes are still being made. We know that they have negative effects on the immune system as well as on neurons, on neuronal function. Okay. And people are also, we can see here, have increased cardiovascular events, kidney, liver, as well as we mentioned osteoporosis. So we almost see this premature aging metabolic disorder syndrome. And this is here just talking about the different antiretrovirals that are available in terms of how they might be contributing to the brain, or this dysfunction in the brain. We don't know. There's Scott Latender has done a lot of work looking at whether these drugs get into the brain, how efficient are they at getting into the brain. It's something that's very difficult to study in humans, right? We can take fluid from the cerebrospinal fluid, but that doesn't always tell us accurately what's happening in the brain parenchyma. And so this is still somewhat an area of controversy. There's also, we know that some of the antiretrovirals, right, as their metabolites, can have damaging effects. So for example, the efavirenz are no longer used because they do cause some neuronal injury. And so from what we know, the brain, I have this on fire. This was the title for my talk, HIV in the brain. And so it leads to this inflammatory cascade where we see the increase of cytokines and chemokines. You have this infiltration of monocytes, which can then differentiate into macrophages. These are the targets for HIV. So neurons are not infected. It is the microglia. So those are the resident macrophages in the brain that get infected, and can the cells become activated, releasing different cytokines, chemokines. This can cause a dysregulation of glutamate metabolism and lead to neurocytotoxicity. And so these are some of the known mechanisms that we have. And so I got interested in, again, because my interest was this host pathogen interaction. So how can we understand how the host responds to HIV? And so I used this older technique, probably you would do microarray or RNA sequencing now, but back then it was a differential RNA display, and it's understanding using our reporter virus, identified the host cell factors that respond to HIV infection, the host cell factors found in macrophages. And it was in this screen then that we identified this very interesting protein called osteopontin. It's a multifunctional protein. It can be secreted. It can also be cleaved. Both halves of the protein are functionally active. It binds to integrin receptors. It has this RGD sequence. It has a putative calcium binding domain in the carboxyl terminus. And just long story short, we showed that this protein is elevated in folks who are HIV infected and have cognitive impairment. We also showed that the protein is expressed in the cells of the brain in these infected individuals, in the microglia and the astrocytes, but differentially expressed in the microglia and astrocytes. And so we don't know, is the protein, what it's doing? Is it having a beneficial effect? Is it a host response to virus infection? Or is it detrimental? Does it help the virus? So our previous studies, using some in vitro studies, we show that when we knock down osteopontin in human macrophages, that decreases virus replication 50%. And it's through a NF-kappa B dependent mechanism, because in the HIV promoter, there are binding sites for NF-kappa B, which is a very important transcription factor, both in the immune system and in the nervous system, actually. And so, again, just to recap, basically, the HIV enters the brain. It's going to replicate in the macrophages and resident microglia, causing their activation. And this release, so interestingly, all of the cells in the brain, so the astrocytes, the microglia, and the neurons, express the receptors for osteopontin. And so we want to understand exactly what it's doing. And so I'm going to scroll through this and go to the next slide. And so we are looking and examining aging and neurodegenerative disorders and chronic inflammation, right? Our favorite organism happens to be HIV, and they can get into the central nervous system. Osteopontin is increased. And interestingly, it's not just in HIV where we see osteopontin increase. Other neurodegenerative disorders, some of you that are working on Alzheimer's disease, Parkinson's, frontal temporal dementia, and ALS, have also shown that there's an increase in osteopontin. And so we think by studying this pathway, we're going to learn something very important about the molecular mechanisms, the osteopontin signaling pathways that are important and that will be common to many different neurodegenerative disorders. And so we're using both in vitro and in vivo systems to get at this question. And so you can see through my pathway, right? I didn't start out with the aim to study neuroscience, but the pathogen, right, has led me there. And I'm working at the interface, and this is the field. It's called neuroimmunology. It's a very exciting field because we used to think in the brain that there wasn't, that it was immune privilege, right, that it wasn't much happening in the brain in terms of the immune response, but there actually is a lot happening in the brain. And so we're working at this interface, the neuroimmune. And osteopontin is very central in this because it has many good functions, but it can also, we think, be toxic, okay? And so we need to, when we have too much of the osteopontin, we hypothesize that it's causing injury. So if we can learn more about it and exactly how to understand its signaling pathways, what are its partners, maybe we can develop some interventions to modulate its activity. And so as other speakers have just mentioned, right, neuroscience is such a fascinating field. I mean, for me, so you never stop learning, right? And so that's very exciting for me. And all of these fields can contribute. I've spoke to a couple of your colleagues today, someone interested in engineering. And he's seeing by being here in the SIP program how he can, in the future, interface possibly with neuroscience. And so I'll tell you about our in vitro model. So I don't know how many of you have heard about humanized mice. Okay, good. Some of you, a few of you. So humanized mice, there's different types, but the one that we are using, because normally mice cannot be infected with HIV. So these are mice that don't have an immune system. We can reconstitute pretty much their immune system by injecting them with CD34 positive stem cells, right? Hematopoietic stem cells. These stem cells will then go to the bone marrow. They will differentiate into the target T cells and macrophages for HIV. And then now we can infect these mice. And so that's just shown on the far right there. This is a flow cytometry diagram showing that when we stain either for human white blood cells or mouse white blood cells, in the animals that get successfully engrafted, we can generate these human immune cells. And so these are studies that are in progress. And what we're asking here is osteopontin required for HIV-mediated neuronal injury and cellular activation? So we are inhibiting osteopontin expression using some small RNA molecules called aptamers. And we are going to then follow these animals and look for measures of cognitive impairment, of neuronal injury, at the end of the experiment by immunohistochemical staining. We can do this in live animals by doing behavioral assessments, so we can study the behavior, right, of these animals. We're also going to look at microglial activation in the brain using PET imaging. So I have collaborators to do this. Mikhail Pletnikov is an expert in behavioral studies, and Marty Pomper is an expert in PET imaging. Okay, and we will. In this first part, we currently are in the first phase of these studies, but we will also repeat these studies and look at the impact of antiretroviral therapy. So I just wanted to show you a brief little video here, but we can study anxiety, right, in these mice. And so this was just a few weeks back. We did our first, it's my first time, doing behavioral studies in mice. And so this is an open field test, and we can measure and quantify how often and how much the mice explore their environment, right? The more anxiety they have, they will tend to stay on the perimeter. Okay? And so, let's see, how am I doing? Okay. And so, as I mentioned, we are, have to work in different model systems, whether in vitro, maybe working with human cells. We can also use rat neuronal cultures, and again, in vivo. And so I just wanted I'm going to flip through some of these slides just in the interest of time, but we had shown that in vitro, we know that the HIV envelope protein is neurocytotoxic to neurons. And so we asked, what is the role of osteopontin? Maybe osteopontin does it accelerate or does it protect from HIV-mediated neuronal injury? And so we could show that osteopontin increasing doses can actually protect neurons from HIV envelope-mediated neurite degeneration. And if we look closer, because the previous study was staining for beta-3 tubulin, which is prominent in neurites, we can also look at specific stains that stain specifically either the dendrites or the axons. And we see that osteopontin can, in a dose-dependent manner, protect from HIV-mediated damage. Okay. And we think that this we've shown that this pathway, when we block osteopontin receptor beta-1 integrin, we can partially reverse this phenotype. There's a lot of literature, so reading the literature, and there was another article, someone was looking in the visual system, and after axonal injury in the visual system, and they showed that osteopontin could actually protect, and that the mammalian target of rapamycin pathway was involved. And so this is a pathway that's been shown to be involved in long-term synaptic plasticity and learning and memory, neuronal survival, and so we've got interested in this pathway and investigated whether it was involved in our system. And so we could show using rapamycin, which will block mTOR signaling, we showed that rapamycin actually reversed the protective effect of osteopontin. So that implicated osteopontin. And I'm going to just skip some of these slides because I just think this wasn't supposed to be so in detail. But we basically investigated several components in the mTOR signaling pathway. And we've shown now, we think that we have some fairly good idea of what is happening. And so we think that the receptor, so HIV uses two receptors, right? The CD4 receptor as well as a co-receptor. And depending on the envelope type, it's either a CXCR4 or it can be a CCR5. In our studies, our envelope protein uses the CXCR4. And what we've shown is that the envelope appears to activate the mTORC2 pathway. So there's two mTOR pathways, 1 and 2. And we think the osteopontin, because we can block its effect using antibodies against beta-1 integrin, is activating the mTORC1 pathway. And we've shown that these other factors are elevated through this signaling mechanism. And interestingly, there's this interaction, though, between the signaling mediated by the viral envelope and the signaling by osteopontin. And so it's the outcome of this interaction that then leads to our phenotype, where we're seeing this enhanced neurite outgrowth. So that's where we are in that story. But it's very interesting, in the nervous system, there are at least 10 different integrins that are expressed in the nervous system, and they have a very integral players in neuron interactions, in neuroglial interactions. And so my fellow, she is now investigating the effect of osteopontin and HIV envelope interaction in hippocampal neuronal cultures. And she's finding very interesting results that we hope to publish soon. And so there were a couple folks I spoke with at lunch. So I think you guys have a good idea. I wasn't sure, who was going to be in the audience, but if you're first generation and explaining to your parents, like, and your family, like, what is you going to do research for the summer, and like they're going to pay you, what is that about? Well, what do you produce? Are you going to sell? Right? Do we sell anything? And so you can tell them that research is very important. It could save lives, right? So. Research, I'm here and I'm still standing because people participated, right, in research. At the NIH, there's lots of clinical trials that are so important, right, because it can impact some of us in this room, our families, right? So my mom, she died from breast cancer when I was like 3, so I don't even remember her. But it's because of research and the passion that people have that allows me to be here, standing here, okay? So as the first speaker mentioned, it's how we respond to adversity and negative events, right? And so it really matters, who our mentors are. So you heard from Dr. Nath. Dr. Nath used to be our divisional director in the Department of Neurology. and this is Dr. Justin MacArthur, who is our director currently, right? These are important mentors that said, okay, when you coming back? Great, they sent me flowers, but like, okay, when you coming back, right? And so where you work and who you work with is so critically important, right? And so, this is me. And so this is my passion. Now you know something about me. This is my first time actually, showing that in public, right? Because when you have some adverse event, when it's real close, it's hard to talk about, right? But it's at a distance now, so I can talk about it publicly. And so I like to inspire students, and when students come to me with good news, this is how I feel, right? It's like, right, athletes, right? I get so excited when they're like, oh, I got this internship, or, what good news that they have. And so, this is where the persistence, so some of it, I don't know where it comes from, maybe some is nature, but I think we can learn it when we have some success, and just realize, right, this is what science and working in the lab, when things fail, right, you get real down, you're like, oh, I don't want to go back there and do that. But when things don't work, that directs you to the pathway where the light is, right? So you have to keep going forward. So my pathway has been maybe a longer range. These are the dollar signs here are the grant monies that I brought in. So it depends on the institution that you go to in terms of how you're supported. So Johns Hopkins is like soft money, so we have to bring in grant funding in order to cover our salary. But again, it's about your personal curve. When I was coming up, it was like, boom, if you didn't, you didn't have a trajectory that was like this in a certain amount of time, you were a failure, And so it's good now, people are taking gap years, right, after they finished their undergraduate. And so it's your personal curve and your experience, what it is that drives you, what contribution you want to make. And so I'm really fortunate. I get to do both the research, I'm trying to do the research and the educational piece, and provide experiences, especially to other underrepresented students, minority students, and these are high schoolers. So we bring in juniors and seniors from high school into the Department of Neurology. They get paired with the faculty. They do research like you're doing for 8 weeks. They also get to shadow the neurologists in the clinic. And we've been doing this for 11 years. This is in collaboration with our department, right? Because our faculty don't get paid to do this, but they're passionate about training and educating the next generation. And so our scholars are getting interested or majoring in neuroscience. And so it's a very exciting time. And so I had put together this word cloud. I think you've heard these words from the prior speakers. And just go for it. Just go for it. And so you remember this picture I showed you earlier in Dr. Atkinson's lab. It just so happened I was at an Understanding Interventions conference in Baltimore. And I saw her. This is Dr. Regina McClinton. She was sitting at the table, and I went and I looked at her. Right. Just got in her face, and I was like, ah! We screamed, we cried, right? But you see, we're both smiling, right? But it all started back there. So your network, right? Make as many friends here. You saw that picture earlier, right? That you can use each other as a resource. It's so important. That's something I didn't do. I didn't know how to do that when I was at your stage. But the network is so important, right? And so these are, this is my postdoctoral fellow, Dr. Farina Mahmood, Thomas Butcher. He just left for medical school. And this list is not exhaustive, doesn't have all of the undergraduate students that have worked in my lab and who have also published with. These are our funding sources. And so, you guys, why we do this is so important. You are our next generation for the next new novel discoveries. And so go forth and be successful. And Thank you so much. So are we going to take questions now? Yes, go ahead. Yes. Yes, yes. So, don't remember that year, but I actually had a miscarriage. So, but thankfully, the place that I work, because it was, this private industry academic kind of center, they were very supportive. And so when I finally did, I got subsequently, because my husband and I, we wanted to have a family, I actually had to go on bed rest. And so that did impact my publication record. And so it did have an effect. But again, I'm standing here, right? And so you have to find a way. You find a way. Maybe it's gonna, take you a little bit longer. Hopefully, you don't have any issue like that. I've seen many successful women who are able to, carry the pregnancy full term and still work. And so it's interesting. I used to think, wow, gosh, look, I have a child now, and I'm still doing all these things. Wow, gosh, what could I have done? Or where would I be if I had these same kind of skills, time management skills? Yeah, I wanted to mention that, right? These non-cognitive skills, your time management, your interpersonal skills, maybe self-advocacy. So all of these things come into play. And so, again, as all the other speakers said, you find a way. You find a way. I used to work on weekends. You can at least write from home. And again, because I'm passionate about it, and now you know why maybe if I push a little bit too hard, because I'm so excited. I'm so grateful to have the opportunity. And so, again, there's no good time. I would say, choose your partner carefully. Because, you both, pretty much it takes two, but you need somebody who's going to, cook, wash your clothes, do all those things, or if you're going to, have somebody do it for you, if your family can help you. So there's all different kind of solutions. And so have very good communication with your partner. Yeah. Yes. Mm-hmm.
Like with excitatory synapses and like cytoskeletal organization. Can you talk about that?
Well, it's very new, new to us, I should say. And so, but the integrins, well, about the synapses, right? You have these many different membrane proteins that come together that are involved in the signaling process. And so, osteopontin, as an extracellular matrix protein, interacts with these integrins. And that can facilitate the signaling that occurs. And so we don't know more than that, but that seems to be where we're leading. And so Farina's work in the hippocampal system, she's doing a lot of knockdown studies using siRNA against beta-1 and beta-3 integrins, really trying to nail down exactly what is happening. Yeah, thank you. Mm-hmm. Okay, thank you.